All right, the last little clip here on environmental factors that will influence microbial growth. Another one is actually whether or not you have other organisms in your surroundings. Do you have neighbors? So your textbook has this diagram, which is a pretty good one, which divides organisms up into different categories that can exist based on whether or not they have any associations with other organisms. Okay, so we have symbiotic types of relationships and non-symbiotic relationships. So a symbiotic relationship is where you have one or more species living together, and that relationship is required by at least one of the two members. So the key word there is required. So there are three types of symbiotic relationships: mutualism, commensalism, and parasitism. So yes, being a parasite is a type of symbiotic relationship, and that might be counter to what you have thought about before, because generally we think about symbiosis as being a good thing, but parasitism is a type of symbiosis because it is required by the parasite. Obviously it's not required by the host because the host is being damaged by that situation. So this is beneficial for the parasite, not beneficial for the host. The host is harmed. Now what about mutualism and commensalism? Mutualism is where both organisms live together and both are dependent and both members benefit. Commensal is where two organisms live together, one organism benefits, the other is not harmed by it. So in mutual, what's a good example of mutualism? So for a long time, once microbiologists discovered that the human body had lots of microorganisms on it and in certain internal parts of the body, like the intestinal tract and the mouth and so forth, a lot of people thought, well, those are going to be commensal types of relationships. The human body's not really benefiting from it, but the microorganisms are. But however, as more research and time have progressed, we've come to understand that a lot more of those microorganisms are in a mutualistic relationship with us. So they're benefiting, but we're benefiting as well. A good example would be good old E. coli that lives down in your colon along the inside lining. And of course it's benefiting by getting its nutrients and moisture and so forth from the inside lining of your colon. But we're also benefiting from it because E. coli can do nice things for us, such as make vitamin K. We need vitamin K. We can't make it on our own, so we have to get it from our diet, from the foods that we're eating, or from E. coli and other types of microorganisms that live in our intestinal tract and produce it for us. Commensalism, now a lot of the microorganisms that live on these may just be commensal, so they're benefiting by getting to live on or inside our bodies, but they're not really benefiting us necessarily. Notice parasites can be broken down into other categories as well. Ectoparasites, endoparasites, intracellular parasites. Viruses, for example, are intracellular parasites. They have to go inside cells in order to reproduce. Ectoparasites are on the outside of the body, endo inside. So you want to take a look at that terminology. And then keep in mind obligate versus non-obligate. A non-obligate parasite is an organism that can live as a parasite in the host, but can also live freely out in the environment without a host. Non-symbiotic types of associations. So sometimes organisms live together, but they don't really have to. A synergistic type of relationship is where the members get together and they cooperate and they share nutrients, but they don't require it. Antagonism is the opposite. So you have two members that are living close to each other, and one of them is being inhibited or destroyed by the other. So some microorganisms do that, and we'll talk more about it later during the semester. Microbial antagonism is thought to be one of the major benefits of our normal microbes, the normal microbiota that live on and inside our bodies. It's thought that by having these friendly microbes living on and inside our bodies, they help outcompete pathogens that might make us sick. So that's called microbial antagonism, and that's thought to be one of the major benefits of our normal, the normal microbes that live on and inside our bodies. So what's the difference between synergism and mutualism over here? What's the key? Because they kind of sound the same when you're first thinking about them. And it really just gets down to mutualism is required by the members, synergism is not. So let's talk about biofilms. Biofilms are a type of synergism that exists between microorganisms. So for a long time, like when I took microbiology for the first time back in the 1980s when I was in undergraduate school, we didn't really talk about biofilms. I think back then the line of thinking was when, with bacteria and other single-celled microorganisms, it was kind of every cell or every man for himself. But we've come to appreciate since then that microorganisms do set up communities that often involve more than one species, and those communities are called biofilms. So this is a synergistic type of relationship, not required, but can be extremely beneficial to the members of the biofilm. All right, so if you may have heard this term used before, and if you haven't, now that you've encountered it in your microbiology class, you're going to start hearing it because it's kind of a buzzword out there these days. So a lot of disinfectants will advertise, for example, that they're really good at dissolving and breaking down biofilms. So what are biofilms? So over here you've got an example surface, and then you've got these microorganisms that are living together in a community. And I remember the capsules, the glycocalyces, the very structured, orderly ones that exist on some types of bacteria. So those types of substances, those sticky, carbohydrate-rich substances, can help groups or communities of organisms adhere to surfaces. And then the members are protected by all those sticky substances that they're producing. So if you think about it, if you've got some disinfectants that are trying to invade the biofilm, they have a hard time doing so. Or let's say this is inside a patient and you're on some sort of antibiotic drug that's meant to kill these bacteria, but the drug can't penetrate the sticky, gooey substances that are surrounding the bacteria. So that's helping to provide a survival advantage to these types of organisms. And biofilms apparently can have pretty complex structures. So what they're showing you here is there are spaces left in the biofilm that allow water currents to circulate through, and that helps make sure nutrients can make it to all parts of the biofilm. Wastes will be carried away. So apparently they can be pretty elaborate types of structures. It's also been discovered that, how do bacteria know whether to live as little single organisms or form a biofilm full of all of these carbohydrate-rich sticky substances? And it turns out that many bacterial species can do something called quorum sensing. So you may know quorum from, if we have this meeting, we can't vote unless we have a quorum. We have a certain number of members present. So what this means is the bacteria, some bacterial species, when they realize that they have lots of neighbors around them, they will begin producing these sticky substances that will generate the biofilm. So they change their behavior by doing that. That's called quorum sensing. So how do they do that? So many bacteria secrete molecules, signaling molecules that are kind of like hormones that you study about in AMP2. And those will attach to receptors on the surfaces of neighboring bacteria, kind of like hormones would attach to a hormone receptor. And if a cell receives enough of these signals from neighbors, it knows, hey, I'm part of a community, and it'll change its behavior and start producing sticky substances that create the biofilm. So that's called quorum sensing when that occurs. So there's a pretty good diagram in your textbook which walks you through the stages of biofilm formation. You want to take a look at that if you haven't done so already. This image from your textbook was made with a scanning electron microscope, a very powerful type of microscope that generates these really beautiful three-dimensional images. And in green on here, now this has been colorized so that you can see the different features better, but the green, those are fibers of a gauze pad. And in blue, you've got these are Staphylococcus bacterial cells. And all this orange stuff you're seeing in here, that's extracellular matrix. So you may remember, hopefully you remember extracellular matrix from when you studied tissues in Biology 201. Those are filler substances between cells. And so that's what these bacteria do. The substances that they produce that create the biofilm that helps protect the bacteria, that's a type of extracellular matrix. And they show you here the quorum sensing and so forth that takes place. Notice also they're calling the first bacteria that come in and attach to a surface pioneers. And they also show that once a biofilm forms, you got all these bacteria in there protected by the sticky substances, that extracellular matrix. Bacteria can break off of there, free-living bacteria can break off, and they can go off and colonize, become pioneers on a new surface and start a biofilm over on a new surface. Or let's say you've got a biofilm somewhere inside the body, then bacteria can break out of that biofilm and spread to another part of the body. So this can actually be a source of infection inside the body. So biofilms have become very heavily appreciated for their medical significance. It's been found that, for example, people who have recurrent ear infections have biofilms along their eustachian tubes. So little kids who have ear infections a lot over here in the inner ear, it turns out that the bacteria may be forming biofilms. So if you got that growing in your ear, and the antibiotic drugs can't get in there and kill the bacteria. This is really gross. This is a cross-section through a Foley catheter that would be inserted into the urethra and bladder of a patient who's hospitalized. So you're looking at it in cross-section, end on, and all this. This was an image also made with a scanning electron microscope. All that crusty purple stuff you see there, that's a biofilm that has formed on the inside of the catheter. That's a big-time problem with Foley catheters. It's very easy for bacteria, especially intestinal bacteria, to get into the catheter and start to form a biofilm in there. And then those bacteria can break off of there and wind up causing a bladder infection, or they might get into the bloodstream and cause an infection of the blood, which can be extremely serious. Some other examples of biofilms, the plaque on your teeth. So we all have bacteria growing on our teeth and inside our mouths. I don't care how frequently you brush your teeth. Hopefully you don't let it get to this extreme though. But any plaque on your teeth, plaque is really a type of biofilm. It's many different kinds of microorganisms that are surrounded by the extracellular matrix they've produced. There's an embedded YouTube video here that you might want to take a look at, and it talks about some research that's being done in North Carolina to try to figure out how can we break apart these biofilms inside the human body that are causing problems with human health. So little kids, for example, who have ear infections and can't get over them, maybe we need to give them a couple of different drugs. One that will break apart the biofilm, and then that will let the regular antibiotic, like the amoxicillin, for example, that a lot of kids take for ear infections, maybe that amoxicillin can actually penetrate and get to the bacteria and wind up killing them. So that would be a pretty good little video clip to take a look at. All right, the next video lecture for Chapter 6, we're going to talk about the study of bacterial growth. And I'm going to hit some highlights from that section in the textbook. The textbook goes into more detail on techniques that are used to measure bacterial growth, and we're not going to really get into a lot of those details. So use your study guide that I've given you in the video lecture here as a guidepost for what you actually need to know from this section 6.3.